So one of the things we're really interested in is trying to decode health and disease signals from the body, right? How do we understand what is happening in our bodies, how to react to them, how do we dive into this really deep? And as we're thinking about this, and I won't belabor this point because Ralph Steinerman really went through something similar, but we're really trying to have a somewhat simple but a model in our heads of what we care about. So we think about a person. They exist in some sort of a wellness state. There's some sort of proper functioning of our bodies. At some point in time, you may have the initiation of a disease process or a process that may have the potential to lead to a disease. There's a cascade of networks that get perturbed. This is all pre-symptomatic, right, that you can see molecularly. You then get over to clinical signs, and eventually, of course, we have various outcomes to that. And so in thinking about this area that we call scientific wellness, the idea is basically most of healthcare is sitting way over here. 97% of all healthcare dollars are spent on care after illness, so you already have a severe problem. And we want to think about, in scientific wellness, how do you focus on keeping someone healthy, understanding those earliest warning signs so that you can predict them and prevent them, okay? And so much of the last 5 years or so that Lee and I have been working together on this has been trying to make that into a reality, build systems for helping us to think about intercepting disease in its earliest stages. And if you want to just think about this really briefly, let's just take a very familiar example of diabetes. Okay, so diabetes is a great example because there is such a thing as pre-diabetes. We know how to mark it molecularly. There are things like hemoglobin A1C. You can look at your resting glucose. You measure how insulin resistant you are and so forth. There's actually a great new paper that shows new markers for diabetes on beta cell dysfunction that comes out of the Providence Network. A place actually off the beaten path called the Little Company of Mary. They actually have this great new paper in the Lancet that shows all this as well. But if you think about interventions, if you have insulin resistance and so forth, you can intervene by diet, you can intervene by going low glycemic, pretty simple kinds of stuff. Once you're down here and you're at late stage diabetes, you're amputating a foot or something like this. And so this spectrum of just trying to move early has huge potential for saving healthcare dollars, saving lives, and enhancing health. So to try to make this a lot more concrete, Lee and I initiated a project together that we called the Pioneer 100 Wellness Project. And this was published last year in Nature Biotechnology, and was really something we did, including a number of people here in the room, that was focused on, in our community, getting 100 people, and it turned out to be 108 people at the end of the day, that came into the program and essentially went through and got many, many different kinds of measurements. And what we wanted to show was that we could get enough information out of those measurements to make it compelling and interesting for people, have an impact in their life through scientific wellness. And the idea is if you can have an impact in their life through scientific wellness, they will stay with you and you can build out the longitudinal data sets that you need in order to predict and prevent disease. And so we started off with a vision of trying to create this for many, many people. Our initial view was 100,000 people that we wanted to get together for doing something like this. And the idea was to generate data that would be dense enough and long enough that you could evaluate the early warning signs for all the major human diseases, so you could develop ways to predict them and prevent them. And so that was sort of the onus of starting this project. And so the assays that we did, so sort of our baseline was whole genome sequencing on everybody. That already gives you millions of differences at the level of SNPs for every individual. We did many labs, so this was about 150 different lab tests that your doctor might run, about 700 different metabolites, about 400 different proteins, and these were measured at 3 times during the year. So we started having dynamic measures. We did gut microbiome measurements also 3 times, so you're getting dynamics during this period. And then much finer scale time dynamics by wearable devices, Fitbits and things of that nature. And so these form 2 sort of purposes. So one is that we mine them in the short run to look for actionable possibilities. These actionable possibilities are informed by previous studies, by interventional trials and so forth, but you have a lot of information, and so you're just pulling as much of that and building a personalized program for someone. And then in the long run, we're building what we call personal dense dynamic data clouds, or PD3 clouds. And the purpose of those, as I alluded to before, is to give us the substrates for trying to understand the early warning signs for disease, and also for building out metrics for understanding wellness. Okay, so that was the foundation. So when this project, which I'm not going to talk much about today, was successful in terms of what we were able to learn scientifically, but also in providing a program that people resonated with and they had a good experience, this led us to then drive scientific wellness forward on two different fronts. So the first is that Lee and I, along with Clayton Lewis, who's the CEO, and a number of great people here in the audience who were involved in the founding team, started a company called Aravale. I won't get into it too much, but it's a consumer scientific wellness company here in town. And the important thing from the standpoint of today is that we have now about 5,000 people who have gone through that program, and between 90 and 95% of them have opted in to allow their data to be used anonymously for research. So all those data come back into the lab. We analyze them here at ISB, and that is part of what powers a lot of the analyses that I'm going to show you today. The second huge element of pushing forward scientific wellness is the wonderful partnership when Rod Hochman, who you heard from at the beginning, approached Lee Hood for Lee to become the Chief Science Officer for Providence. And this has given, and ISB has now become a part of the Providence family. And we're so happy about that. It's been really an amazing journey for us. And now that gives us access in a system that is servicing millions and millions of patients all throughout the Western United States, the third largest nonprofit healthcare system in the country. And so what that does now is we've launched a series of clinical trials where we're using the same dense dynamic data clouds, not only in well populations, which we are doing, but also in Alzheimer's disease and multiple sclerosis. We're looking to launch a big effort in pregnancy and so forth. So there's many, many facets to this. So both through the consumer wellness side at the population that comes in sort of as they are, as well as healthcare focuses that comes in via the Providence partnership as well. Okay. So I'm going to tell you a number of little vignettes. I'm not going to go super deep into the science on each of these. I'm going to try to keep it fairly high level and talk about the future, since that's the point of today, and do that in a timely fashion. So the first thing, though, I do want to give you one little tidbit about the kinds of things that we learn about the health intervention for people. So I'm going to use some data here from individuals who have gone through the Aravale program. To acknowledgments at the beginning, analysis from Andrew Magis, fantastic former PhD student in my group who's now Director of Research at Aravale, and Jennifer Lovejoy, who's run this whole program and put it together. And I'm just going to show a tiny snippet from this. So one of the things that you can do is to pull polygenic scores to inform different blood measures that can be related to disease, many different areas. And the idea here, this is one of the criticisms of consumer genomics, is that individual variants are statistically significant, but usually they have small effects, and so how meaningful they are for an individual person, certainly debatable. Polygenic scores make that a lot better because by aggregating the effects of many different genes, you start to explain much more of what's going on, and that can become meaningful for a person on an individual basis, and I'll show that in an example here in just a moment. And we can build these genetic risk scores for all kinds of traits, all kinds of diseases, and so forth. So here's one. So LDL cholesterol, the bad cholesterol, everyone's sort of familiar with this. And what you see behind me is polygenic risk scores that have been calculated for all the people who are in the Aravale program. And they've been binned into 5 groups, so quintiles. And what you can see here, if I'll get out of the way, is this low quintile to high. And so what this says is that based on your genetics, you can see how linear this is. Based on your genetics, you can predict sort of what is likely to be your LDL cholesterol level. So there's a very strong genetic effect to this, okay? So one of the things that we were interested in, and we hypothesized a few years ago, but we didn't have hard data on, was that if you take an intervention, say to try to lower your LDL cholesterol by lifestyle intervention, that it's probably easier to do so if your genetics predicts that you should be low than if they predict that you should be high. So what we did is actually took individuals who are in the Aravale program who have the same measurement for LDL, okay? So these are people that in the current healthcare system would be treated as exactly the same. You know, you have your LDL, you compare it to the average of the population, and you treat, okay? So what we have here instead is that we actually showed for people who have the same LDL level, what changes they went through when they went through this scientific wellness program. And what you'll see is that those whose genetics predicted that they would be high had a much harder time lowering their LDL cholesterol. You'll see that they did lower a little bit, not statistically significantly, because you can see the error bars cross zero. But for the people who had space there where their genetics said they should be low, they actually moved very significantly, okay? And so what this showed, we think for the first time, quantitatively, although we had expected that this might be true, is that you can predict and personalize for individuals what are the areas that your genetics will allow you to improve the most by lifestyle intervention, okay? All right, so that's all I'm going to show for data on that segment, but I just want to make a few points on implications for the future. So one, healthcare systems in the future are not going to treat blood measurements against population averages without looking at the genome. That is going to be viewed as archaic within a decade. I'd love to say sooner. Healthcare moves sort of slowly usually, but within that time frame, because everything should be personalized to your genetics that tells you specifically, where you should operate. Another big implication for the future, and we haven't worked this all out yet, is that in some of these cases, it may be that your genetics predicts what is a proper homeostasis for you. In other words, the ranges that are used also should be personalized to the individual. And in some cases, the genetics will just be sort of a bad hand. Maybe everyone, maybe low LDL cholesterol is good for everybody. But in other cases, it might be that the genetics tell us different levels are better for different people. And we'll sort that all out for different measurements at different times. And you can imagine that in the future, personalized health plans driven by your genetics, the comparison of where you are to what your genetics says that you should be at, and then you could give personalized ROI maps, essentially, for anybody that says, these are the actions you should take. You're most likely to get benefit from these because your genome is telling us you have a lot of room for improvement. Your body actually wants to be there. It will work with you rather than against you. And so those kind of things we're very excited about. So that's implications for the future, number one. Okay, second point. So I now want to shift and say we have all these data clouds, so we can also do a lot of statistical analysis to try to understand them. And so one of the areas that we're really interested in is the microbiome. It's a very new area, and everyone's sort of scrambling to try to understand health implications of the microbiome, as are we. And so these data clouds we've generated actually give us one of the best data sets in the world, I would argue, for trying to understand the microbiome, not because it's the largest microbiome cohort in the world, but because it is the microbiome cohort where we have more other kinds of data than basically, because we have the genome, all the data I showed you in the Pioneer 100, now amplified over 5,000 people. So we have a lot of this information. All right, so I'm just going to show you some of the beginnings of this analysis. So there's this huge relationship, which I won't belabor, between gut microbiome and health. It's just starting to be sorted out. These PD3 clouds are an interesting tool for that. And so one thing that we've done right off the bat is show, and this is work, I showed their pictures, I should mention this work done by Noah Rappaport, Tom Womansky, and Sean Gibbons, who's a great new microbiome faculty member here as well. And so, in essence, what we're able to show is that there's actually a strong ability to predict microbiome alpha diversity from the blood metabolome. And I'm going to talk a little bit more about that. And so quantitatively, we get, you can see the statistical significance here, 10 to the minus 56, reasonable sort of correlation. And we can do that primarily off of a pretty small set of metabolites, most of which are human microbiome co-metabolites, so things that they have partial metabolic pathways for across both the microbiome and human, and you can see sort of the makeup of the molecules that are there. So a couple things that are really interesting is that, so we tested this and we said, okay, is this predictive ability that we have for an individual's alpha diversity, so just how diverse is their microbiome, which is related to health. How well can we predict that from metabolome? Is it robust across different conditions? The answer to that was yes. So when we looked at people, so if they had abdominal pain, so this is now looking at disease conditions within the Aravale population. So abdominal pain, you can see that M Shannon, which is our predictor from the metabolome and the actual Shannon diversity, and you can see that in every single case, these things move together. And so it picks up across many different disease conditions what's happening in a person's microbiome. So one thing that we think that you can do out of this is, for example, you can build a predictor from a blood analyte that tells you whether or not you're likely to have dysbiosis, and maybe you should go get your microbiome checked out because it's shifted into a really low diversity state. And you can put that into a routine blood test, right? We could roll that out. If you look at the variance explained here, so here's clinical labs, you can see that the maximum, which is BMI, which is there for comparison, everything is worse than that. So basically all clinical labs that are used in hospitals, or at least all of them that we measure, which is a pretty good fraction of them, don't tell you anything about the state of your microbiome. Okay, now the new biomarkers, or the notion of using this deep metabolomic surveys, now you're representing nearly half of the variance. So you're able to make much, much more predictions. So the metabolome contains a lot of information about your microbiome, your clinical labs, not so much, okay? At least about its diversity. So another thing that was really interesting here is that this relationship between the metabolome and the microbiome was not affected by antibiotics. In other words, if you took antibiotics, it shifts your microbiome diversity, but you see it in the metabolome. So that predictive accuracy holds up, okay? Sex didn't affect it very much, neither did age. So it was very robust. There's two things, though, that do affect it pretty significantly that we're interested in. One is inflammation. So if you have really high inflammation, this relationship between your metabolome and the microbiome will break down. And the other area that this is in, and I want to be quick here, is related, and that's if you are if you have extreme obesity. So if you BMI over 35. So obesity, as it's defined, obesity level 1, it still holds, and you still have a good predictive ability. But if you shift into this BMI over 35, so you're sort of extremely obese, if you get into that regime, again, this breakdown between the metabolome and this relationship between the metabolome and the microbiome breaks down. And there's some interesting things on this. There was a really interesting paper in Cell Metabolism we actually reviewed, but it's come out now, that went through this and tried to differentiate between the healthy obese and unhealthy people that weren't as obese. And that was all centered around analysis of metabolism. And so we think that some of the mixed signals in this field that we've seen from this relationship might come from not separating out this extreme obesity from kind of the middle obesity. So we only find it at the extreme edge, but this relationship breaks down, and so it says, okay, there's something different happening between your metabolome and your microbiome in that severe of a case. All right, so talking about the future, so implications for the future. So these PD3 clouds really give us a wonderful platform for trying to understand health. And the thing that I'm gravitating towards now is I actually think that analyzing the blood metabolome, especially the exometabolome that comes from the microbiome, is going to be our quickest, best way to understanding what is a healthy microbiome. I think it's going to turn out to be a lot more fruitful in a short term than studying the microbiome itself. Because the microbiome, you can have many different microbiomes that are interfacing with your body, and they have to deliver certain things into your body. And so I think these data are going to allow us to understand via the metabolome and its interface back to things that we understand, say in clinical labs and so forth. That is going to be the rubric, I think, to understanding what is a healthy metabolome. And this is what Noah and Tom and Sean, who's collaborating with us, are all working on as well. Okay. All right, I have two more stories I really want to tell you. Okay, so the third story here then is work that's been done by John Earls, also with some great input from Laura Heath. And this is getting at not just trying to use these data to understand disease, but to understand health. So this is a first pass at what Lee has called a metric for wellness. And we think there's going to be many of these where you're trying to understand more about a health state. So this is one where what we're looking at is biological age, right? So other people have done this kind of work. There's epigenetic clocks that Steve Horvath, for example, down at UCLA has done to try to predict age. And so what we're doing here is taking the molecular data that we have in the PD3 clouds. One thing that's really nice about these data is you have so many facets to it that it's easy to go back and see what are the drivers for someone looking older or younger than they would appear molecularly by their chronological age, okay? And Lee likes to point this out, right? This is his 80th birthday, but it's only his 65th birthday by biological age, so many, and I won't show the data on this, but on average, people in the Airveil program by this measure are getting slightly younger year over year, so we're pretty certain that we're going to be able to push Lee younger over year. That's our goal. Okay, so here though, what we have is, so we have our biological age from dense phenotyping. Essentially, I won't get into all the details, we use something called a Clamara-Duval algorithm. In essence, that's to try to make it so that 1 year of chronological age is roughly equivalent to 1 year of biological age, so that slope is meaningful. Essentially, you just build a regression from part of the data so that you build a model of what, given a given a PD3 cloud, how old would we predict this person is, and then you apply it into a test set and see how that does. So the first thing that we wanted to do was to say, is there any validity to this notion of a biological age as one sort of rough measure of health status that someone could key in on? And what we found is that if we took a whole bunch of different disease conditions, in every single case, everyone that didn't cross the line here at 0 went in the direction of looking older. So this was kind of just a sanity check. So everything that that in this data set of the top 20 conditions represented in the Airveil data that we thought of as bad, all major biological age go up, okay? So that was just sort of a sanity check to say, okay, it seems like it reflects in some sense worse health in a way. And that's what you see here. And the kind of things that are captured in this biological age calculation, it's really related a lot to metabolic health, to inflammation, bioaccumulation of toxins comes up. Things like lead are very obvious because they come into your body and your body doesn't have a good way to get rid of them, so they have a tendency to accumulate. And so those kind of things go into the score. Okay, so flipping to implications for the future. So this biological aging is a first metric for wellness in the sense that you're trying to understand a person's general health state. There's a lot of caveats to that because, and I'll show those in a moment, where we're going to shift and talk about the detailed analysis related to disease. We do think that in the future, there needs to be a lot more study of wellness. And one thing I'll mention just very briefly is I think the genome has been missold to the public in a pretty massive way. So the view that I have is that the general person on the street, when they hear the genome, the picture is it's some sort of crystal ball that tells you how you're going to die. So this is, don't get your genome sequenced, it's all scary stuff, right? This is kind of the view of the person on the street, I think. And in reality, the genome should be viewed as a key to understanding your life. And one of the things that I think is so key about this, and I'm going to use an example of this that I learned from Jeff Bland here in town, which I really like. But we have a tribe of Native Americans here, and there's papers out and it talks about how they have diabetes genes. But they, of course, don't have diabetes genes. They have genes that are adapted for the lifestyle that they, lived and thrived on in the plains. And so those genes are they're not adapted to be diabetes genes. They are proper for a certain kind of environment and a certain kind of lifestyle. And so learning about your genome is important and essential because it helps teach you elements about how to interface with lifestyle and environment and so forth. And so that's why we really think that the study of the genome should not be solely focused on disease, although that's where most of it has been, and we've done a ton of that as well, but also around something that's related to health. All right, last story. So the last phase then is I want to talk about data that we get because we can get it before, during, and after disease transition. So I already talked about this, so I won't talk about it again, but just to remind you, right, wellness, disease initiation, preclinical, and that's what we're going to be looking at. So I'm going to show you some examples of this. So these are all examples of people who have come through the Aravale program who have unfortunately been diagnosed with some sort of a disease, but these are also opportunities to try to understand or look at transitions and try to figure out how can we stop these kind of things from happening. So I'll start with the first example, and this is actually the very first example we looked at, so it's kind of our canonical one from the beginning. So we refer to this woman often as Eve in these, an anonymous name in these studies. But basically here, this was a woman who was diagnosed with stage 4 pancreatic cancer, which, as you know, is a very grim diagnosis. My father-in-law actually died from pancreatic cancer a couple of years ago. It's a nasty disease. So Bert Vogelstein estimates that if once you've got your diagnosis, you've probably had the disease for 5 years. So she probably had this when she came into the program. So she was diagnosed at the beginning of 2017, but we had 4 blood draws prior to that, so up to about 20 months pre-diagnosis. And if we look at her 4 cases, we go back into the proteins, and there's one protein in particular. There's other signals as well, but this is the strongest one that pops up, and she's an outlier in this protein. So we look it up, we're like, what is this? Well, it's made primarily in the pancreas, related to pancreatic cancer. It's in the Notch signaling pathway, and you can see that she's a major outlier on this. Now, this is a million miles away from being a biomarker or something like that, right? There's tons of things, and I'm going to get to that in a moment. But there's at least a real possibility that this was a signal for her. And so what we've built now is a system that will monitor for all of these outlier events. So we can characterize for any person very rapidly, here's every analyte that you have that's in a very unusual state relative to the well population that we see. Okay? And I'll talk more about that in a moment. You can also do this for networks. So another thing that happened, this is just an example of one of the networks. One network that became very perturbed in this woman was the Tnf Signaling Pathway. And I won't get into the details of how we calculate this. We've actually adopted an older method of ours. Noah Rappaport has turned it into an N of 1 method, so we can apply it now in an individual patient, but we can analyze that. But if we look at these kind of networks, we took a set of signaling networks out of KEGG, and we just reduced down to 9. So we're just looking at 9 pathways or 9 networks here. And we reduced to 9 just because we had good coverage in our proteomics data for these 9 and that were reflected in the blood. But what was fascinating in her case is if we do visit 1, so this is 20 months before diagnosis, she looks totally normal. All of her pathways, we would say, are absolutely normal. Now, if you step forward 6 months in time, now one of her pathways seems to be in an unusual state. You go to the next visit, now 2. Just a few short months before diagnosis, now 5. But you can see that those, so this is us, in essence, capturing what we've said theoretically, which is this disease-perturbed, cascade of networks, and it comes through, and now we see that she has moved on that. And so even if we don't have specific biomarkers for this kind of approach, we can start to look at the dynamics for someone and say, something unusual is cascading in your body, right? Do we understand what that is? Is that a warning sign? And so forth. So I'm just going to tell you one more thing here, which is this happened just a couple of months ago. So we had a woman who was coming through the program, and we were doing analysis of her data clouds. And in this case, one thing that popped up that was which was alarming, was alkaline phosphatase. And if you know anything about alkaline phosphatase, it is a marker for bone metastasis. Okay. Now she doesn't have a cancer diagnosis, so it pops up. So she's given this information to take to her physician. Her physician sees her and says, "Oh well, this is a worrisome sign if you had cancer, right?" But says, "Oh, you're on this drug. You're on this drug, valacyclovir." He says, "Oh, valacyclovir is known to increase alkaline phosphatase, so no big deal, go home." Well, with these kind of data, one of the things you can do is you can ask this kind of question immediately within So it took us 5 minutes to do this. John Earls did it in 5 minutes. So we did this. We get these distributions of everyone who's on this drug. So we just happen to have 55 people on the drug. Well, there actually is no difference between people on this drug, and we actually went in the literature to see what evidence there was for this shift upwards for people on this drug. Turns out it was quite weak. And even if you believe it, it's like 10%. It's a minor shift. She's 7 standard deviations above. Anyway, she gets back into a different doctor, goes through and sees this, and in fact does have bone metastasis, which she is now being treated for. Now, one of the things, and I don't have time to get into this at all today, but we think this kind of information needs to be right at the fingertips of doctors. And in fact, we have a new company that we're starting called Consilience, which I won't get into, but we've got a few of our co-founders here in the room. So Lee and I are co-founding a company along with John Alden and Scott Paris. And one of the goals of doing that is to get this kind of information right at the fingertips of doctors, where you have an ability to deliver insight to them in a short run. So we're really excited about that. That's another thing we're looking at for the future. So just to sum up, and I'll be done, is what we're trying to do now is to build a new paradigm for diagnostics that's different from basically everything that we've done before, and we've done a lot of this. So typically you have your case control design, you get a bunch of cases, you get a bunch of controls, you compare them, and you figure out what the differences are, and you try to go through and develop biomarkers. And we've actually done that to commercial success a couple times. And so there's an important path there. But this is going to be different. Because right now, since we're looking longitudinally and we're focused on the wellness state, there is no case control. Okay? So what we do is we define everything based on the wellness state. What is usual for health? Then what we do is on an N-of-1 basis. So for every individual, we have a readout of here's everything that's unusual. That's a measure in U. Every analyte, every network, the relationship in the network, and it's all personalized. And then the hard part is this, which is then what we're doing is a functional analysis of that individual difference. And Jim's actually going to talk a little bit about this, I think, this afternoon, which is rather than just do a statistical analysis, we're going to try to mechanistically and functionally understand what is it about what we're seeing in these divergences. Does it give us clues as to what is happening? Now, and I call this the context interpreter, for lack of a better word. We'll try to think of a better one eventually. But basically what that does then is it lets us get at what would be truly personalized medicine. Because this kind of a system would let you make a diagnosis, understand what was happening in that person, even if you have never seen that pattern before. So it's truly personalized. So it's a different path. It has its own challenges. It has its own difficulties compared to the traditional path, but it actually would deliver truly personalized medicine. All right, with that, let me thank especially Lee, who has been so inspirational and wonderful partner throughout all of this, and all the great members of the lab. I thanked basically everyone along the way except Gustavo Glusman and Gil Ullman, who were both huge parts of the Pioneer 100 project as well. And with that, I will be done. Thank you.